lurking in the dark, spreading like wildfire, causing thousands of infections. Mold, a looming threat to public health, asthma, ABPA, hypersensitive pneumonitis, and even cognitive impairment such as depression. Mold-related diseases affect millions. Spreading by airborne spores, indoor mold infestation reaches a prevalence of around 22% worldwide, with a formidable level of pathogenicity. Locally in Shenzhen, where the climate is high and humid, mold contamination accounts for 30% of all cases of childhood asthma. In the US, the number of asthma cases attributed to mold is 4.6 million people, with a cost of $3.5 billion. Mold is everywhere. Where moisture exists, mold exists. With minimal nutrients, mold spores grow into mycelia, extending hyphae deep into the building wall surfaces, tissues, everything. Beyond a health issue, it damages building structure, destroys historical relics, leading to an annual loss of $40 billion globally. Current solutions include physical brushing, household chemicals, and oxidizers. Brushing removes only the mycelia, leaving the spores that can quickly grow back. Household chemicals, such as soda and vinegar, are also ineffective. Oxidizers jeopardize health and bleach surfaces, leaving people with increased risk of infection and extra expenses. It seems that molds are unstoppable, but in fact, it is not. Aiming to wipe out this pervasive public health problem from the source, GreatBay-SCIE 2025 developed a brand new mold eliminator, ArMOLDgeddon, targeting different structural components of mold cells. Different from other fungicides, ArMOLDgeddon annihilates active mold growth in a safe and effective way.
Our engineering focus on developing a multi-component antifungal system targeting key structures of mold cell walls. Since chitin and glycan form a rigid protective barrier, we combine specific chitinases and glycinases with human lysozyme and geraniol for synergistic degradation and inhibition. Experiment followed a framework of expression and activity evaluation, with the SDS page and breadth-full assay confirming protein yield and DNS assay measuring enzyme activity. To target chitin, the major structural component, 4 chitinases RMV-CHI, BCCHA1, GRX-CHIB, and PRCHIA were selected based on their strong chitin degrading ability and proven antifungal activity and expressed in E. coli. 3 yielded good expression, with BCCHA1 and RMV-CHI exhibited high specific activity toward colloidal chitin at 37 degrees Celsius. GRX-CHIB showed no activity, while PRCHIA formed inclusion buddies. To improve performance, optimization was employed to redesign these 2 chitinases. Through the enzyme activity assay we developed, PRCHIA3 and 5 were identified as soluble variants with measurable activity in the supernatant. For glycans, we employ multiple glycinases with synergistic specificities. BGLS27, BGLU1, and FLGLU30 each interact with different glycosidic bonds. They were all successfully expressed in the supernatant and show hydrolytic ability. BGLU1 possessed the highest catalytic efficiency of 532 U per milligram and was able to react most of the substrate within 10 minutes. Given that beta-1,3 glucans account for up to 80% of total glucan content, BGLS27 was also optimized for yield and activity. Furthermore, being inspired during an interview, we found that alpha-1,3 glucan also plays a crucial role in the cell wall stability. Therefore, we expressed AGL-EK14. Unfortunately, we didn't obtain the expected band and require further investigation. We also attempt to express in human lysozyme, which exhibits antifungal effect through its cationic properties and oxidative mechanisms. Using P. pasteuris as chases, protein expression is attempted via genome integration to His locus using a linearized plasmid. Transformation is ongoing and future experiment is underway. To enhance surface retention and utility, enzymes were fused with the CBM2 module from Bacillus, increasing adherence to materials like wall paint and filters. The additional CBM not only cloned the PET slices strongly, which persisted after rinsing, but also improved the catalytic activity of both the chitinase and the glycanase for around 20%. Geraniol, a monoterpenoid, disrupts fungal membranes. In the E. coli with PMVA pathway constructed, we integrated the geraniol synthesis module made up of a GGPPS2 and an OBGES to allow the GPP produced to be directly converted into geraniol, boosting yield and efficiency. By truncating the first 65 amino acid of OBGES, we also achieve a 62.8% yield increase, reaching 0.157 gram per liter. Reaction of enzymes with geraniol added confirmed that it does not impact enzyme activity. To address geraniol's volatility and hydrophobicity, we produced gamma-CGTase to convert starch into gamma-cyclodextrin, encapsulating and forming stable inclusion complexes with geraniol. Collectively, these modules target mold from different facets, forming a multi-layered antifungal system. Most of our enzymes were verified, while some performances remain limited by structural constraints, so we employ modeling to guide rational redesign.
With the aim of improving enzyme activity, soluble yield, and stability, we carried out optimization through a modeling approach, focusing on the catalytic domains of two kinases GLX-HIB and PRCHIA and a beta-1,3-gluconase BGLS27. Our general workflow comprises four major steps: structure acquisition, design space selection, sequence redesign, and dry evaluation. As a central theme to our improvement pipeline, the third step, sequence redesign, is realized through deep learning-based tools, including protein MPN, which targets improved protein folding fidelity, and ligand MPN, which targets higher catalytic activity. Crucially, the enzymes contain conserved regions and catalytic motifs, which upon modification could render the enzyme inactive. These regions should be handled with caution. The two kinases GLX-HIB and PRCHIA have solved protein structures deposited in PDB databank, while gluconase BGLS27 has none and is thus modeled by AlphaFold to obtain the structure. After that, we carried out ligand docking and iterative multiple sequence alignment through HH-splits to define active site and find the top 50% conserved residues, with the rest set as input for MPN redesign. For BGLS27 redesigned by ligand MPN, we noticed that the top 50% conserved region encompassed the entire active site and binding site, and thus adopted a more comprehensive approach, modifying the entire active site for one batch and the entire binding site for the other. 384 sequences and 180 sequences are generated by MPN for kinases and the gluconase respectively. The generated sequences are modeled by AlphaFold and assessed based on a matrix containing PRDDT, Rosetta score, solvent accessible surface area, etc. Since BGLS27 has all of its active site residues selected for ligand MPN redesign, we considered that an extra scoring term would be necessary to assess the effect, and include a glide docking score in the screening matrix. All these criteria ensure that the variants with improved characteristics are favored. After screening, 6 best-performing variants are selected for each enzyme as candidates for wet lab validation. Wet lab results show that 5 of the 6 PRCHIA redesign variants exhibit good soluble expression with highest yield of 262 mg per milliliter, contrasting inclusion body expression for wild-type protein. Among the 5, 2 candidates displayed kinetolytic activity, highlighting the success in our enzyme modeling effort and setting our redesign pipeline as a good starting point for future iGEM teams. The redesigned variants of GLX-HIB and BGLS27 are not satisfactory with all expression in inclusion bodies. Exploring the reason, it is noticed that most PRCHIA variants have significant increase in SASA value, which may have contributed to their success. In the future, we plan to investigate other tools to continue our modeling journey.
To make sure our product truly meets real-world need, we build our human practice around one goal: turning ArMOLDgeddon from a lab concept into a responsible, socially considered solution. We engaged over 20 stakeholders across 5 progressive stages and 12 cycles, guided by our 4A principle: aspiration, action, analysis, and adaptation. We started by understanding the problem. Surveys across China show that 79% of people have battle household mold, frustrated by an ineffective cleaning method and the pungent odor left behind by chemical product. Doctors and the Shenzhen CDC confirmed that mold causes serious infections and allergies. Their feedback led us to focus on safety, long-term stability, and pleasant odor, choosing non-toxic enzymes and natural terpenes like geraniol, and launching public education to raise awareness about mold hidden risks. Next, we identify our key users: hospitals, restaurants, and household. Housekeepers and the restaurant owners told us that current products are corrosive, smelly, and short-lived, while hospitals needed non-irritant, biodegradable solutions safe for vulnerable patients. We even tested existing products ourselves and experienced firsthand the eye and throat irritation they cause, confirming the need for a safer enzyme-based alternative. This insight guided us to designing hardware prototypes like a long spray head for high ceilings and anti-mold AC filters for humid environments. To ensure feasibility, we consulted experts across microbiology, engineering, and industrial design. They help us enhance enzyme efficiency with AI-based modeling, add alpha-glucanase to target mold spores, and ensure our approach stays safe and environmentally friendly. We also explored enzyme immobilization techniques and powder formulations to improve shelf life and simplify use, bringing ArMOLDgeddon closer to real applications. To bring our solution to market responsibly, we interviewed investment experts and bioproduction specialists. We developed a sustainable roadmap, spray drying for stable powder product, high cell density cultivation for mass enzyme production, and microbial fuel cells to treat waste sustainably. This ensures ArMOLDgeddon can be affordable, accessible, and eco-friendly. In our final stage, we expanded our impact. We reached out to cultural relics preservation experts to protect fragile heritage materials and the Maldives Hotel to adapt our design for global humid climate. We also published the ArMOLDgeddon booklet, an illustrated guide that teaches families and communities how to understand and prevent mold safely, bridging science and daily life. Through each reflection cycle, we listened, learned, and evolved. Our human practices transformed ArMOLDgeddon into a project that is not only scientifically innovative, but also socially responsible, sustainable, and human-centered.
Dear human practice stakeholders, a mold-affected patient confronted us with unexpected questions. Why is E. coli? Isn't it dangerous? What is synthetic biology? We've realized her voice echoed a wider community, exposing a deep divide between cutting-edge science and public awareness. To bridge this gap, there's only one path: education. We set out not just to teach, but to connect, starting with the young. For primary students who think in shapes and stories, we turned genes into colorful puzzles and invited them to draw their own microheroes. They didn't just learn about DNA. They imagined and created with it. Middle schoolers knew the terms, but not the real-world meaning. So we brought Symbol to their hands, extracting banana DNA, solving plasmid puzzles, visiting a real Symbol company. Over 95% left not just understanding synthetic biology, but feeling it. As learners grow, so do the challenges. Our high schoolers began with offline lectures and an iGEM club, then delved deeper through newsletters and online classes to improve geographical and temporal diversity for public school students. To enrich the experience for students in international schools, we brought something new: Symbol Night, featuring a lively GDCA and PCRC session and ethical debates. And Symbol Orienteering, a hands-on collaboration game where participants team up to complete missions at each checkpoint, uncovering the principles of genetic bricks along the way. And for the broader public, we met them in everyday context. Through public talks, board games, and social media, we even invited parents into the lab to dye fabric with bacteria. Step by step, engineered organisms transform in their minds from something abstract into something tangible, safe, and sustainable. Our educational approach grows like a living tree, rooted in 4 core principles: thorough, hands-on, interest-guided, and communication. These roots sustain everything we do. We move fluidly between theory, experimentation, and ethics, listening as much as teaching. The public has shaped conversations on real-world problems and future aspirations, while we've developed 3 types of teaching tools and 19 reading resources. These efforts have taken roots across 27 Chinese provinces. We don't simplify science. We bring people closer to it. Through trust and participation, synthetic biology becomes tangible. That's how we've built not just understanding, but a community imagining the future with us.
By incorporating effective lab design, education activities, integrated human practice, entrepreneurship, and innovative measurement techniques, ArMOLDgeddon not only addresses integral issues with scientific precision, but also promotes human health, sustainable innovation, and community well-being. Through these efforts, we make tangible contributions to multiple SDGs, demonstrating how synthetic biology can create real impact in both everyday life and environmental sustainability. ArMOLDgeddon, science with empathy for every breath we share.